Over the last 15 years, Alzheimer's research has shifted its focus from the production of amyloid beta toward the mechanics of clearance failure. We scrutinize the decay of the sleep-gated glymphatic system. We measure receptor-mediated efflux and the dysfunction of astrocytic aquaporin 4 channels. These are all legitimate routes for waste removal, but they describe a hydraulic drainage network. A drain is useless without the fluid running through it. To model the brain's waste management by studying only removal is to study a bathtub while ignoring the tap. That fluid is the product of the choroid plexus. This is a gland of high metabolic intensity, manufacturing half a liter of cerebrospinal fluid every day and pushing it outward against steep osmotic gradients. A perivascular drainage network cannot clear waste if it is starved of fluid. It requires continuous upstream hydrostatic pressure and constant turnover to maintain the chemical sink that pulls amyloid out of the brain. The choroid plexus operates as a border organ, positioned at the precise interface between the peripheral blood supply and the fluid bathing the central nervous system. This location grants it three distinct offices. It is the source, providing fluid turnover, the sieve, manufacturing native amyloid chaperones, and the sentinel, serving as the immune gateway to the brain. In tracing how these functions decay, we have to be careful. Mechanistic enthusiasm in Alzheimer's research frequently outpaces reproducible human evidence. We will audit the organ's three offices, mapping the specific failure points and rigorously grading the literature supporting each claim. This grading allows us to determine the organ's true role, whether it is the primary initiator of the disease, a passive victim, or an active amplifier of the collapse. At the standard blood-brain barrier, the vessel walls themselves form the seal. The choroid plexus is different. Its vessels are porous, and the barrier is thrown up one layer out, at the epithelium. This metabolically active sheet allows the plexus to act as a secretory gland, manufacturing a protein called transthyretin. Transthyretin is a native chaperone. It binds freely floating amyloid beta, prevents it from aggregating, and assists in pulling it out of the cerebrospinal fluid, handing it back toward the blood via LRP1 receptors. Bidirectional mouse genetics provide the causal link here. Overexpressing the human transthyretin gene protects against the disease, while silencing the endogenous gene accelerates the pathology. Clinical data corroborates this, showing that transthyretin levels in the cerebrospinal fluid drop significantly with age and in patients with Alzheimer's. Because protection scales with the availability of this protein in vivo, the failure of the sieve office earns an evidence grade of strong. We turn now to the second office, the source of the brain's fluid tide. Secretion depends on cellular polarity. Sodium-potassium pumps on the fluid-facing membrane extrude ions to generate a gradient that draws water through aquaporin channels. In Alzheimer's, the tall cuboidal cells of the plexus atrophy and flatten. The basement membrane thickens, and the expression of ion transporter and mitochondrial genes falls. Direct ventricular infusion studies suggest that cerebrospinal fluid production rates in Alzheimer's patients are roughly half those of controls. This halving of the tide starves the downstream clearance system. However, there is a known confound. Cerebrospinal fluid production tracks with brain metabolic rate, which independently declines in the disease. It is difficult to determine if the plexus is failing or simply responding appropriately to a less demanding brain. Consequently, the source failure earns an evidence grade of moderate. The final office is the sentinel, where the plexus functions as an immunological gateway between the body and the brain. Genomic analyses shows that the aged choroid plexus develops a chronic type 1 interferon signature. This program is typically mounted against viruses, but here it suppresses the organ's reparative and immune trafficking functions. The epithelium also responds to systemic inflammation by releasing extracellular vesicles into the fluid. These vesicles carry inflammatory microRNAs that activate astrocytes and microglia throughout the brain. While the mechanism is vivid, translation has proven difficult. Attempts to therapeutically reopen this gateway using PD-1 checkpoint blockade failed to replicate in independent multi-laboratory studies. The biological description of this interferon shift is robust, but the lack of reliable therapeutic translation demotes the sentinel office to a grade of speculative. Beyond cellular mechanics, we can observe the behavior of the choroid plexus in living patients using MRI. These observations reveal a physical paradox. While an atrophying secretory organ should shrink, The Alzheimer's choroid plexus actually enlarges. This structural scan highlights the contrast in volume. Inflammatory cell infiltration and lipid accumulation drive the spatial growth, rather than an increase in functional capacity. This structural swelling correlates with cognitive decline and the ApoE4 genotype, linking the disease's primary genetic risk factor to this border organ. Imaging confirms the organ is involved in disease progression, but these cross-sectional correlations cannot distinguish whether the swelling is a cause or a reaction. Completing the validity ledger allows us to place the choroid plexus within the etiology of Alzheimer's. Tau pathology in locus coeruleus precedes clinically identifiable plexus failure, suggesting the organ is likely not the disease's primary initiator. However, because plexus secretions like transthyretin actively shift the trajectory of amyloid pathology, the organ cannot be labeled a passive victim. The data supports the role of a permissive amplifier, a border node where degradation removes chaperones, weakens the flushing tide, and opens an inflammatory gate. Because choroid plexus enlargement occurs before cognitive decline, it serves as a non-invasive preclinical biomarker for trial enrichment. This role also suggests a therapeutic pathway, developing stabilizers for native transthyretin tetramers to restore the brain's lost chaperone safely. We cannot model the hydraulic collapse of the Alzheimer's brain by staring only at the drain. To understand the flood, we must also interrogate the tap.